Good day all. Welcome to today's session on monoclonal and polyclonal antibody production. I myself is Dr. Ganga. So what is an antibody? Antibodies are immunoglobulins which are glycoprotein in nature. They have a combating site for antigen and these antibodies are produced by plasma cells in response to stimulation of B cells by a foreign antigen. Antibodies are glycoproteins which is aroused against a particular foreign antigen which comes in our body. When you see a typical structure of an antibody, you can see two major structures. That there is a upper portion is called a FAB portion and the lower portion is called a FC portion. So the FAB portion is the antigen binding, that is fraction antigen binding position. Here, each antigen which is coming and binding to the particular antibody is depends on the amino acids composition in this particular area. And this is called a C-terminal domain. Here there is a COOH group, and here it is a amino group. And this C-terminus, this help in the fixation of this antibody onto the cell surface. So there are two major chains which is made, this antibody is made up of two major type of chains, heavy chain and light chains. And in between we can see a disulfide bond, and here in the neck portion we can see an hinge region. So this is a typical structure of an antibody. So when an antigen and antibody interact each other, they interact by means of certain forces like hydrogen bond, electrostatic bond, hydrophobic interaction, van der Waals forces, etc. Antigen and antibody reactions are always reversible in nature. When you talk about antigen-antibody interaction, you have to know two terminologies like affinity and avidity. So what is affinity? It refers how a specific combination site on antibody is attracted to a specific paratope. So if an antigen and antibody have multiple epitopes, multiple region of sites, so and which specific region in which an antigen and antibody bind, that is affinity. Affinity is required for an antigen and antibody to bind each other. Whereas avidity refers the overall interaction between antigen and antibody complex. So how they interact each other and forming a complex, that is the avidity. The sum of all affinity together. So how much ever, say for here we have number one antigen, always sometimes it is, say for example, number one paratope. So it is interact with the number one antigen with the number one paratope. Again, we have number two antigen interact with the number two paratopes. So that is the affinity. Each one interact with other one is affinity, whereas the avidity is the sum total of this affinity is called avidity. So what are the major functions are immunoglobulins? The most important function of antibody are neutralization, opsonization, killing by oxidation, agglutination, precipitation, antigen-dependent cellular cytotoxicity, antibody activate complement system. These are the major functions against microbial pathogen, that is the immunoglobulin function against the pathogen which is interact in our body. So there are different types of immunoglobulin in our body. We call it as isotype of immunoglobulins. There are major 5 types of isotypes of immunoglobulin like IgM, IgA, IgG, IgE, IgG, and IgG and IgD. So IgM, IgA, IgG, IgE, and IgD are the major immunoglobulin isotype which is present in our body. So the epitopes is the immunologically active region of an immunogen which can arouse an immunogenic reaction. So this epitopes is present on the antigenic determinant. This is the antigenic. If you take an antigen, the entire Suppose if you consider antigen as a ball, the entire ball it cannot be act as an antigen. Only a specific portion, maybe the tip of the ball, one side of the ball, can be act as an antigen. So exactly, the epitopes is the antigenic determinant region of a particular antigen, and there are different types of epitopes can be seen in a particular antigen. So these antigens are recognized by uh, immunoglobulins and can raise some immunological reactions. So a antigenic molecule, any antigenic molecule that can evoke an immune response are called immunogens. All immunogens are antigen, but not all antigens are immunogens. So, what are the influencing criteria that influence pathogenicity? Is always an antigen which comes in our body should be foreign to our body. It's called foreigners, and it should have a specific molecular weight. Lesser molecular weight and higher molecular weight cannot be interacted each other. Then it should be a chemical complexity. Then genetic constitution of the host, that is genetic makeup of the host. Route of administration, that is also very important. Route of administration, how, in which route it can be administered, that is very important. So if you look into this picture, we have different types of epitopes. As you said, this is a one type of epitope, this is another type of epitope in a same kind of suppose this is a pathogen, it itself have different types of epitopes in the body, and for each specific epitope there will be a specific kind of antibody. So it's look like the shape itself. So for this is specifically meant for this kind of epitope, and this antibody is specifically meant for this type of epitope, and this antibody is specifically meant for this type of epitopes. So epitopes are nothing but antigenic determinants, and the binding of epitope, the antibody site, antibody part of epitope, that the opposite part of epitope is called the paratope. The paratope is the antigenic antibody part which comes in bind with the epitope. So we already known about what is an epitope. It is a key and lock interaction. Epitope and paratope is look like a key. You cannot open a key if you don't have a specific key. You cannot open a door which if your key is not rightly fit into the keyhole. So similarly, the antigen antibody reaction should be taken place if it is rightly placed. If a paratope and epitopes are rightly placed each other. Now we are moving on to different two types of antibody which can be possible, monoclonal and polyclonal antibodies. First, we are coming to monoclonal antibody. Mono means single, which is one. Singular type of antibody produced by harvesting purified cell line derived from a single B line within the immunity and allowing them to bind to one unique epitope of an antigen. So this is a kind of antibody which is specific to a unique epitope for a single epitope, single type of antibody against a single unique type of epitopes are called a monoclonal antibody. So monoclonal antibodies can be produced. There are two major scientists, Kohler and Milestone. They got a Nobel Prize for this particular work. So monoclonal antibodies are produced by immunizing injection of an immunogen into an animal, often a mouse, against a specific antigen. So if you this is to a particular antigen or particular antigenic determinant, a particular type of antibody can be produced. So after the immunogens are caused an immune response, the B cells from the spleen are removed. For this purpose, we are taking a typical single type of antigen and inject into mice, and the mice will produce a similar type of antibody against this particular type of antigen. And those once the B cell is getting activated, those spleen cells has to be removed. That is the first step of monoclonal antibody production, that is injecting a specific antigen into a mouse and try to evoke an immune response there, so that by B cells which is capable to produce a single type of antibody is being removed from the spleens. The spleen containing B cells is removed. There are some B cells which can be later during the we call it as cancerous B cells, which can able to multiply forever or proliferate forever. So we call it as immortal B cells. That is that is called carcinoma. We call it carcinoma cells. So there are two major types of B cells, which is taken. We call it as the B cells, which is lymphocyte B cells, which is having an immunogenic response, and another type of B cells, which is turns to myeloma cells or cancer cells. They have the ability to multiply an n number of times. So, and they have taken the advantage of myeloma cells is it is having the n number of multiplication, whereas the normal B cells, though it can able to produce antibody, it is having a short lifespan. So the scholar and Milestone thought, why can't we fuse them together? So the character of the myeloma cells, that is the immortality of the character and the antibody producing ability of the B cells can be fused together, and both characteristics can be brought into one cells. So they try to fuse the cells, and by somatic hybridization methods, and they produce hybridoma cells, and the technology was called as hybridoma technology. So it is a method of producing a large number of identical antibodies called a monoclonal antibody. So hybridoma technology is a technology which is produced by Kohler and Milestone in producing a similar type of antibody from a single type of cells, that is called identical antibody from a similar clone of cells is called a monoclonal. That is single clone. It is called a monoclonal antibodies. So hybridoma is an immobile cell derived from a fusion of B lymphocyte with a myeloma cell. So hybridoma was a hybrid. Hybridoma is a immortalized cell derived from a fusion of B lymphocyte from a myeloma cell. Myeloma, that is cancerous B cells. So for that they took a selective medium called a hypoxanthine aminopterin thymidine medium that only hybridomas can grow, and unfused myeloma cells cannot grow in that medium, because the unfused myeloma cells, when the presence of aminopterin, the de novo synthesis pathway is getting reverted, so that the myeloma cells cannot be grown alone in the hypoxanthine aminopterin medium, whereas the unfused B cells, and the B cells can grow, but they are having a limited period of time. Whereas when they try to fuse this both cells in a selective medium called a hypoxanthine aminopterin thymidine, called short form is called an HAT medium, the fused cells were thrived in that medium, whereas unfused myeloma and unfused B cells were started dying off. So the mechanism of growth hybridoma cell is a selective medium where B cells containing a gene called hypoxanthine guanidine phosphoribosyl transferase, but myeloma did not have this gene. So the B cells, the normal B cells, which is able to produce the antibody, that is called splenocytes, they have have a particular gene called a hypoxanthine guanidine phosphoribosyl transferase gene, but it was absent in the myeloma cells. When they fuse together, the few hybridoma cells that got the B gene, the HGPRT gene from the B cells, and the fused hybridoma cells started growing in the HAT medium. So for what is the peculiarity of this gene is HGPRT gene is necessary for replication of the B cells. So HAT media only have the HGPRT positive, the HAT media with HGPRT positive enzymes. That means the cells which is having the ability to produce HGPRT enzyme was thrived in that medium. The HPTR negative was just thrown off or they got died. So it was a selective medium. So the hybridomas later which was produced were able to grow continuously, and they could able to produce antibody continuously. So they were later screened for the production of desirable and monoclonal antibody, that is, they were screened for the production of antibodies, and they were continuously, that single cell which was able to grow continuously as well as able to produce monoclonal antibody was selected, and then they were grown massively in tissue culture. So that is a single clone of cells that is having the ability to produce single type of antibody. So they were purified, and the monoclonal antibodies were separated later after the purification procedures. So once a monoclonal hybridoma made stable, and we could able to repeatedly produce antibody from the particular cells, that was acted as a renewable source of antibody. So and they ensured whether the previously produced antibody is in the first batch is equal to the second batch of antibody, and every batches of antibody they used to check simultaneously, and they produce if it is the first batch of antibodies is showing the similarity between the second batch of antibody, third batch of antibody, then they will release it as a monoclonal antibody. This monoclonal antibody can be also produced in direct injection of hybridomas, in the peritoneal cavity of mouse, in order to create tumor that secrete a fluid rich of antibody called ascites fluid. So this is the this hybridoma can also produce by induction of tumor in mouse cells, and this tumor can secrete a fluid which are rich in antibodies called ascites fluid. So that is an in vivo condition, and that is for each time you have to sacrifice a mouse. But in case of monoclonal antibody, it is a cell-based assay, so there is no need of sacrificing an animal. So it is more advantageous than the in vivo hybridoma technology. So this was the schematic representation of what I told you. This is we are taking a specific antigen and injecting onto mouse, and there is and injecting onto mouse. Then we are removing the spleen cells from the spleen. We already have a cell, that is myeloma cell culture in our hand, and we are taking the myeloma cells which is in the cell culture, and we are trying to fuse this spleen cells with the myeloma cells, that is immortal myeloma cells, in a medium called a HAT medium. This is the HAT medium, and we are fused with the polyethylene glycol, a compound called polyethylene glycol. It is we are fusing it in the polyethylene glycol in presence of HAT medium, and we are trying to grow that into that particular medium, and those who have thrived, that is hybridoma, will grow further, and the rest of them, those who are not hybridoma, that unfused cell, will die soon, and we can select those hybridoma later, and we can use it for the monoclonal antibody production. So moving on to the advantages of monoclonal antibody productions are they can produce large quantities of identical antibodies. They can batch to batch, there will be a homogeneous set, first batch, second batch, third batch, whatever batch you take, similar kind of antibody can be obtained. They have high specificity against a single antigenic epitope and reduce the risk of cross reactivity. So it's always have an antigen antibody reaction with a specific antigen and specific antibody. Hence, there won't be any cross reactivity. Cross reactivity means a single antigen can react with the multiple kind of antibodies, because the antigen there are mimicking each other can react with the n number of antibodies. So that cross reactivity can be reduced by me using monoclonal antibody. Assays requiring quantification of protein level. So when we assay this monoclonal antibody, the quantification or qualitative method, the assay levels will be very good and efficient in affinity purification. So they can be very efficiently, we can do an affinity purification with the monoclonal antibody. So some of the, though we are telling about the advantages, there are few disadvantages. They are much more expensive, and significantly it needs more time, and storage conditions are much more demandable, and less strong detecting protein denatured state. So there we cannot be able to detect the denatured protein. Uh, it is ideal for requiring quick capture of target protein, more sensitive to pH. So buffer condition should be very, very important. That it will act only at a specific pH. So more susceptible to binding changes when labeled. So while labeling with the radioisotope or fluorescent label, that binding capacity can be changed. That is some disadvantage of monoclonal antibodies. But otherwise, it is effectively used in treatment therapies as well as diagnostic, etc. Now we are moving into poly. There we learned about mono means single, poly here, poly means many or multiple. So this is a collection of antibody which are grown from different B cells. This make them capable of recognizing multiple epitopes on the same antigen. So in a single antigen we take, it is must a single, suppose we are taking a single pathogen, Staphylococcus, it must have multiple epitopes. So multiple epitopes can be recognized by a same kind of, or on the same antigen can be recognized by a multiple kind of antibody. So this collection of antibody which are grown on different B cells, so though they are grown on different B cells, but they can identify a multiple epitopes of the same antigen. So here also there will be there is a procedure for the production of polyclonal antibodies. Antibodies used for research and diagnostic purpose of obtained by injecting the lab animals such as rabbit with a specific antigen. For this purpose, always we need a lab animal. That is one of the disadvantages, because always we have to sacrifice an animal for this purpose. So that is always what is a kind of rule of thumb is we have to inject and specific antigen. Suppose we have to raise an antibody against a particular virus, so we have to take the viral suspension of viral tissue masculinization and directly inject into animal. Sometimes the neuronal viruses like rabies virus and all, sometimes you have to put the virus into the neurons, that is directly inject to the brain or hippocampus region and allow them to multiply. So when you inject the particular antigen into that antibody, I mean, animals, there are different routes of infection, peritoneal cavity, we can do the epidermal type of injection. So intravenous injection, there are different routes of administration of this particular antigen. But once you inject an antigen, the animal immune system will produce high level of antibody specific for the particular antigen. So they will start producing antibody against that particular antigen. And this is not a one-time injection is not possible, and subsequently we have to mix this antigen with some adjuvants, and that adjuvant is having it will increase the immunogenicity, so that along with the adjuvants we have to inject this antigen by booster dose. So suppose we have taken inject this animal in a 1 week 0 day, that is suppose we are injecting today, you have to inject the same antigen with the same route of injection at after a 2 week. After that, another booster dose in the 3 weeks. So we have to expose the immune system with this antigen and gradually increase the production of antibodies in the particular animal. These antibodies can be harvested in an antiserum, which is a whole serum can be collected from an animal following exposure to antigen. So these antibodies can be present later. After 3 weeks, the animals has to be it has to be bleed. We have to take blood from the animal. There are different procedures for that also. Heart injection can be directly taken. The ear vein can be cut, and we can allow the animals to breathe. Anyway, it is a torturing for animals. So the antibody can be harvested in an anti-serum. So it is obtained in the blood is taken, then the serum is separated, the immunoglobulins will be there in the serum. So the most antigens are complex structures, multiple epitope. They result in the production of multiple antibodies in the lab animals. So there be since it is a multiple epitopes, there will be multiple production of antibody. Suppose we are injecting a rabies virus, it is having multiple epitopes. And unlike monoclonal antibody, we are selecting a single type of epitope. Here, in an organism, there will be multiple epitopes. So, entire organism is injecting means for all the epitopes, there will be different type of antibody would be produced. Probably it is must be for the rabies virus, but the epitopes will be different. So, it is called a polyclonal antibody. It responds to typical response to infection by human immune system. So the animals, like as we said, we are taking the antiserum, and this antiserum containing antibodies from multiple clone of B cells, and each B cell respond to specific epitopes of the antigen. So antiserum obtained from this animal will containing antibody against antigen artificially introduced laboratory, but also containing antibodies to other antigens. But it's not only for that antigen, because we are not directly, we are not taken a single virus and not injecting. We are just grinding the virus tissues. Suppose, as I said, suppose a liver infecting virus, hepatitis C virus, or as a brain infecting or a neuronal virus called a rabies virus. We all taking liver tissue, we are just grinding and injecting. We cannot take a single virus and inject. We are taking the tissue of the virus which it harbors and we are injecting into animals so that the antigen of that particular tissues can also be activated against in a particular mouse or rabbit. So for this reason, the antiserum must be first purified to remove other antibodies using antibodies for the research diagnostic assay. So we have to be very careful so that the unwanted antibodies which is possibly raised during this time should be removed by certain specific technique. So in this monoclonal polyclonal antibody production, the same we are injecting the antigen into rabbit. The antigen activate the B cells inside the rabbit and the multiple. So here we can see 1, 2, 3 epitopes. So more than one B cells can produce antibodies against each epitopes. So multiple B cells can be produced. That is plasma cells producing polyclonal antibodies. And there will be a memory cell. So during the second exposure to the same antigen, there will be more amount of antibodies will be produced by the B cells. And as I said, there will be, in the case of polyclonal antibody production, the injection is in booster doses, 1 or 2 or 3, sometimes up to 5 times we will inject, depends on the antigen or depends on the requirement of our work. So once with the antigen is injected, there are two types of cells will be produced. One is plasma cells, other one is memory B cells. So plasma cells immediately produce antibody, and memory cells will stay there. And when they get the second booster dose, so immediately they will start producing, proliferate and producing large amount of B cells, which is capable to produce particular antibody. So this antibody which is produced is that secreted antibody will be there. We are taking the blood out of the particular animal and obtain a serum. So we are taking the serum, we are allowing to settle down the blood cells, and we are taking the anti-serum, finally the anti-serum from that. And then we have to purify this anti-serum, and we will use this polyclonal antibodies for the various purposes. So advantages of polyclonal antibodies are they are inexpensive in nature, they are easy to store, quick to produce, ready to use under 4 months, but it is a low cost production when compared to the monoclonal antibody production. They're highly stable and tolerant to pH. So when compared to the monoclonal antibody, they are quite stable in nature. Their high affinity as antibodies bind to more than one epitope. So here it is high affinity will be there because though it is miss one epitope of a particular organism, there will be other epitope that there will be antibody against other epitopes will be there. So that affinity will be more than amplify signal from target protein even low expression level. So even if it is a low expression level, they can amplify signal from the target proteins. Now, we have to compare the major important aspects of comparisons of polyclonal and monoclonal antibody. So monoclonal antibody is very expensive in terms of production, whereas in the case of polyclonal antibody, they are much inexpensive. Next is a long production time. So in the case of monoclonal antibody, it requires long production time, whereas polyclonal antibody is rapid production time. So it's more specificity in the case of advantage of monoclonal antibodies. They are much more specific than the polyclonal antibody, where it is very less specific in the case of polyclonal antibody. So monoclonal antibody recognizes a single epitope of an antigen, whereas recognize multiple epitopes of an antigen in the case of polyclonal antibody. The monoclonal antibody production is continuous and uniform once hybridoma is made. So they are continuous and uniform in nature. So in case of polyclonal antibody, different batches vary in composition. There will be different batches, they vary in composition. The few host species, it is produced in monoclonal antibody, whereas multiple host species are an option in the case of polyclonal antibody. And monoclonal antibody is an homogeneous antibody population. So it's a single type of antibody will be produced and will be present in the case of monoclonal antibody, whereas polyclonal antibodies are heterogeneous antibody populations. So these are the major comparison of monoclonal and polyclonal antibodies. So I said polyclonal antibodies, some of the disadvantages is they are cross-reactive in nature. So there will be highly variable. The first batch will be having maybe a type of antibody production, and the second batch may vary because the animals varies, the injection times varies, the feeding time, uh, so everything matters because it is produced in a live animal. So the conditions of the animal which surveys, everything varies the production of antibodies. Affinity purification of the serum will typically be required to minimize the cross-reactivity. There will be a affinity purification procedure is really required, and that is quite expensive in nature. That is for the reaction to minimize the cross-reactivity. So that's the end of today. Thank you. Thank you for your patient listening.